Welcome everyone to Iris Biotech's first online workshop. True to our slogan, Empowering Peptide Innovation, we are keen on knowledge exchange and getting in contact with scientists all over the world, which is why we are starting this Iris Biotech workshop series. Today, the main topic is click chemistry, surrounded by some brief introductory information on our refresh company logo and the company itself, on the occasion of our 20th company anniversary. My name is Karin Rustler. I'm working as a project manager at Iris Biotech, and today I will guide you through this online workshop. If you wish to announce your participation on social media, please feel free to use the hashtag Iris Workshop Series, which is also displayed in the Microsoft Teams chat right now. To have a pleasant experience for all participants, I would like to begin with some technical guidelines. To ensure the best possible audio and video quality, all participants are automatically muted and video camera is switched off. Another important information is that the whole live event is recorded. Thus, if you enter late or miss anything, please check on the bottom left side of your Teams screen if you are watching the recording or if you are live. You have the possibility to pause the video, to rewind, or jump back to the live session. In addition, the recording of the workshop will be uploaded to our homepage afterwards. Questions can be submitted by using the Q&A chat of Microsoft Teams. You can enter your name or submit your question anonymous, just as you prefer. We will frequently check the questions, publish them, and submit them to a respective speaker. If you have technical queries, you can also submit them in this Q&A chat, and our technical assistant will then connect with you and try to solve the problem. Initially, the questions are just visible for the organizers and will then be published to the audience. So don't worry if your question takes a short moment to appear. To try out the Q&A chat, we would now like to post a question in the Teams chat which you could answer. So you should see the question, when was Iris Biotech founded, within the chat. And we are checking if there are answers coming in. And yes, you should see there is already one answer. And the correct year, as mentioned, is 2001. So this is also why we are currently celebrating our 20th anniversary. So hope you are now all familiar with this Q&A chat. And before we start, I would like to give you a quick overview on today's agenda. We will start with a short company introduction by our CEO, Steffen Tropic, including the presentation of our new logo, followed by a general overview on click chemistry by our project manager, Dr. Markus Weishaupt. Our external speaker, Dr. Anne Zemella, will provide detailed information on her research at the Fraunhofer Institute about novel opportunities for protein modification by click chemistry. As mentioned, questions will be collected throughout all talks in the Q&A chat and are forwarded to the speakers. Of course, you can also get in contact via email or phone call after the workshop. So one last note from my side, if you accidentally might close your Microsoft Teams window, there's no need to panic. You can just re-log in by using the same link. And as the workshop is also available as recording, you won't miss anything. So let's start with a quick company introduction. Therefore, I would like to introduce to you one of Iris Biotech's CEOs, Steffen Tropic. Steffen studied business administration and received his Master of Business Administration from the Otto Friedrich University in Bamberg. As part of the International Business and Technology Management Program, he spent 2 semesters abroad at the University of Technology in Finland. Afterwards, he supported his father Oskar Tropic at Iris Biotech. And in 2010, Steffen joined the company, became executive assistant, and from 2015 on, he was appointed CEO by the shareholders and took over the management of the company. Since then, he controls the fortunes of Iris Biotech together with his father. Thank you, Steffen, for being with us, and we are looking forward to your presentation.
Karin, thank you very much for this kind introduction. It's a pleasure to have you all here on board for our first workshop here on 2nd of June. And when I was asked to give a little talk here about 20 years of Iris Biotech, it was not that easy to think about a certain topic, because to bring something within a time frame of 10 minutes, that's always quite challenging. But one thing what we all the time had, nevertheless, we didn't speak about that was empowering peptide innovation. And then I thought, okay, it might be quite interesting, because the one or the other knows already our standard PowerPoint presentation when we visit each other about the history. I thought, let's go a step even further back and show to you how we started Iris Biotech, or how the story was behind the foundation of Iris Biotech. So if you all remember the time 2000, when the internet boom was and the IT technology was coming up all around the corner, there was another thing. It was this biotechnology, this new thing booming up. Millions and billions of dollars were spent. And during that time, also, the field of peptide synthesis was very novel. And during that time, a former business partner of my father, they talked to each other, and then they came up with the idea, Oscar, what about a new company for amino acids, for this whole life of peptide synthesis, for this peptide chemistry, to have starting materials for this field? Because both companies received already some inquiries for little tiny quantities, which are in fact normal for universities, so like 1 gram, 5 gram, 25. But both companies were selling this in kilogram or even larger scale, and not functionalized and so on. So there was an idea, and the question was there a market? And when this business friend asked my father asked him, well, what is peptide chemistry? Because my father is a businessman, and then he got an explanation. He went to internet. He went to internet by modem. So all the people who are a bit older, they know this nice scratchy noises. And in fact, he saw that this is really a potential market, but he also knew immediately that he has not the right skills to enter this market, that he and his partner needs a person who fits to that and who has the expertise. And in fact, that was then also the time when Thomas joined the company. It was in 2002. So because already in 2000 and 2001, there was an outlook, okay, we need a certain person. And Thomas was already known in the market. He sold peptide synthesizers. So in fact, he knew the customers. We had some suppliers in the backhand. And that was also one key persons to have. And another person who joined then later on was Annick van Koolen. You all also know her. And they both started, in fact, then also after the foundation, the marketing and the sales for our products. Often when you start in new companies, business partners, banks, or whatever, they ask for a business plan. Well, in that case, there was none, because there were two entrepreneurs, and in principle, they had a rather good idea what to do. And you know, entrepreneurs, they do not make too much plans. They just try it, and they do it, and they get a good feeling for certain things. One thing was also the financial background, and one rule was keep the costs low. In that way, that to spend money for a good person and for equipment which was needed, but not more. And during that time, it was quite good that, for example, the administrative work and things like that were handled by one of the companies of one of the shareholders. What was also quite crucial, and this is also always part of a business plan, is to have the network and relations to the customers. So this was then also given by Thomas and his expertise, but also by the shareholders. They also had certain contacts, but also the suppliers. If you do not get a good product from the suppliers, your product will also have always difficulties. And then for sure, you need good partners like banks, business angels, venture capitalists, or whatever. So in our case, the banks were quite helpful. And then another part, which was also very crucial, was that these two entrepreneurs, the two founders, they got trust and confidence in the market. They were known. And when they started this new challenge, this new journey with a new company, that the business partners who got in contact, they knew, okay, these partners are behind, we can trust on that company. So in fact, then finally, the foundation was done in 2001, late February, but business really started later on. And it started, in fact, in 2001, in second half, very slightly, and then 2002 was the real first serious business year. But one question always comes up during the foundation: how shall we name the company? And then this business partner of my father said, well, Oscar, your daughter is called Iris. Why not call the company Iris Biotech? And my father, he loves his daughter, but he would never call a company just because of his daughter. So he also thought about, okay, what is the kind of connection to biotech? And Iris, well, you know, all the flower and the part in the eye, which we all have. So in these three elements, you also found and find still in our logo. So the name, quite prominent, so quite easy. So here you got then the flower, and here we had the eye with the iris. Empowering peptide innovation means for us also, besides giving you new compounds, new technologies, it somehow has also to fit the layout of the company. And therefore, when we then celebrate, or when we got 20 years old that year, we thought, okay, now we have also to work on the corporate design. And then we asked the designer, and then he came up with an idea. Here in the middle, you still see the iris, the flower, and the round part is still only the part of the eye of the iris, so very reduced. And the daughter is still the daughter, so iris is still iris. So in fact, these were the two parts, and that's why we changed now the logo, and that's a bit the story behind how it is done and how these all elements fit together. You can have a look on our website. There's a little movie which you can see. What changed, or in which direction developed Iris Biotech all over the years? So in principle, the product portfolio increased to around about more than 7,000 compounds which we have. I listed here also all the different categories in which we're active. And if you remember the one or the other meeting when we were seeing each other the years before, one sentence or one question which always came up from the one or the other from you was, what's new from Iris Biotech this time? And even if we saw each other each second month or even each month on all the different shows, we were able to answer, well, now that's new, or that's new. And that's something also what we try to keep up is to give you all the time new compounds, new possibilities, but not only for the lab scale, we also provide this for the bulk quantity so that we increase the quantities according to your projects. And nevertheless, and also quite an interesting part is custom synthesis, as we have a quite high level team who do quite an amazing chemistry, where the one or the other customer already said, well, honestly, we did not believe in that someone could do the chemistry, and honestly, we were really proud to hear that because we were able to deliver the compound in the quality which was defined. What is all behind know-how and competence? Well, in fact, it's not me, it's the team. And here you also see the whole team, which is currently active for Iris Biotech. And where we're also very happy on is that in principle, the team is expanding. The one or the other person is coming and joining the company, but all the older ones, they also stay with us. So that's good, and this gives us also a lot of know-how and knowledge. We are active on the latest developments in the field of peptide synthesis. We have a lot of know-how. We do the marketing of products and technologies. We have and we use our worldwide network so that we can also do the requested and the needed things what your project requires. And we also are aware and are able to do the pharma business, which has its own rules, where then at a certain point chemistry steps back in the priority and qualification and documentation and approval of the production is much more important than the latest chemistry. To summarize, well, what's also new, we got new technologies. So linkerology, that's a term which we created. That's also quite interesting. We are active in the drug delivery and in much more fields, but today it's a focus on click chemistry. Thank you very much for listening to me. If you have certain questions, please feel free. And Karin, thank you very much. The show is back to you. And Markus, good luck.
Thank you, Stefan, for presenting the story behind Iris Biotech and also the new design of the company logo. So with this quick introduction, I would like to continue with the next agenda point, an introduction to click chemistry by our project manager, Dr. Markus Weishaupt. Markus studied biochemistry at the University of Bayreuth and obtained his PhD at the Max Planck Institute in Berlin in the field of solid phase organic synthesis. In 2015, he joined Iris Biotech as a project manager. And by the way, for the ones of you who already downloaded it, Markus was also responsible for the relaunch of our new brochure on click chemistry, which you can download for free on our homepage. So Markus, thanks for your valuable time. Thanks for being with us, and please go ahead with your presentation. Markus, sorry, you are still muted.
So now I should be unmuted.
Perfect, yeah.
Very good. Thank you, Karin, for your kind introduction. And I would like to start off the scientific part of this workshop with a short overview on click chemistry before we go to Dr. Zemella for a more in-depth exploration of certain exciting aspects of this technology. So what is click chemistry? There are various possible definitions of this term. One of them being that it is a category of bioorthogonal and highly efficient reactions. At IRIS Biotech, we categorize click chemistry into 3 generations. The first generation is the classical copper-catalyzed azide alkyne 1,3 dipolar cycloaddition developed by Meldal and Sharpless. The second generation is the strain-promoted azide alkyne cycloaddition developed by Bertozzi and her group, published in 2004. While the third generation encompasses inverse electron demand Diels-Alder reactions. In the context of click chemistry, that means mainly tetracenes reacting with some sort of strained cyclic alkene. The applications of click chemistry are manifold. They range from drug development, lead discovery and optimization, to molecular biology, polymer chemistry, surface functionalization, material science, and many more. So the first generation, the copper-catalyzed azide alkyne cycloaddition, is characterized by a high thermodynamic driving force, meaning that it goes to completion rather rapidly. So how does it work? In the lower left-hand side of the screen, you can see a simplified representation of the catalytic cycle. First, copper forms a pi complex, the triple bond of the alkyne, followed by deprotonation, which affords this complex here. Then the azide coordinates to the copper, generating this copper azide acetylide complex. This activates the azide and allows for reaction of the terminal nitrogen with the alkyne. Then cyclization occurs, followed by dissociation of the product from the catalyst and reprotonation. And the catalyst is regenerated for further reaction cycles. Notable is the high selectivity of this reaction for the 1,4 substituted isomer. An advantage, of course, is that many azide and alkyne building blocks are readily available. As already mentioned, the reaction is bioorthogonal. However, one has to bear in mind the cytotoxicity of copper, which limits in vivo applications. The second generation is the strain-promoted azide alkyne cycloaddition, meaning the reaction of an organic azide with a strained cyclic alkyne. This reaction, in contrast to the classical version, is copper-free, meaning it can be applied in vivo. A nice thing about this variant is the tunable reactivity and lipophilicity of the cyclic alkyne. You can see a collection of available or known cyclic alkynes in this graph here in the lower left-hand corner of the screen. However, one must also note the heavy pattern coverage of this class of compounds. So lastly, third generation of click reaction is an inverse electron demand Diels-Alder reaction between a tetracene and usually some sort of strained cyclic alkene. Usually transcyclooctene is used. So inverse electron demand, what does it mean? A normal Diels-Alder reaction occurs between an electron-rich diene and an electron-poor dienophile. Inverse means that the diene is electron-poor and the dienophile is electron-rich, which of course directly affects which of the frontier molecular orbitals take part in the reaction. The third generation click reaction is copper-free, just like the strain-promoted azide alkyne cycloaddition. However, in contrast to the sp-AAC reactions, it is also very, very rapid. So it is ideal for biological systems and applications such as live cell imaging. Another nice aspect is that you can tune reactivity and solubility of the tetracene by varying the substituents. on the tetracene, and also there are many alkene reaction partners available with different reactivities. And you can also use, for example, cyclic alkynes as reaction partner. This reaction is also irreversible due to a loss of nitrogen that occurs. And one last advantage is the very limited pattern coverage. So to sum up, there are 3 generations of click reactions. All are highly efficient and bioorthogonal. There's a huge number of variations for many different applications. There are many tunable parameters such as reactivity, lipophilicity, steric bulk of the reaction partners, etc. And click chemistry is suitable for both chemical synthesis and molecular biology. If you want to learn more about this topic, we have a brand new booklet. It's called Everything for Click Chemistry. You can find it on our website, iris-biotech.de, under Knowledge Base, Downloads, Catalogs and Brochures. So with this, I would like to thank you for your kind attention, and if there are any questions, I'd be happy to take them.
Thank you, Markus, for giving this talk. And as you just mentioned, so if anyone would like to ask a question, please just use the Q&A chat to submit your question. There is already one question. So is the copper catalyzed click reaction also feasible in vivo?
Well, if you observe certain precautions, it is. There are certain compounds that are able to complex the copper, and thus they attenuate the cytotoxicity of copper. So THPTA is one example. Those are tris triacyl compounds. There are some variants, some more water soluble, some more lipophilic, but yes, there are possibilities.
Okay, so thanks for your answer. So far, there are no further questions, so I would say we just continue, and in case any one of you has further questions, just get in contact afterwards. So after our introduction, we would like to go on with today's highlight and check out some applications of click chemistry. So it's a real pleasure to introduce to you Dr. Anne Zemella, Research Associate at the Fraunhofer Institute for Cell Therapy and Immunology in Potsdam, Germany. Anne studied biochemistry at the University of Potsdam and obtained her doctoral degree for research in the field of protein labeling and tRNA amino acid tRNA synthetase pairs. In 2019, she started her postdoctoral research at the Fraunhofer Institute. Anne, it's a real pleasure to have you with us. The stage is yours, and we are looking forward to hearing about your research.
Thank you for the kind introduction, and I want to thank the organizers for inviting me and for giving me the opportunity to present the results of the department Cell-Free and Cell-Based Bioprotection. As you have heard, I'm working at the Fraunhofer Gesellschaft, and nowadays the Fraunhofer Gesellschaft has nearly 30,000 employees, and we are working at 75 institutes and research units that are located around Potsdam, and we have some international institutes. And in 2020, we had a finance volume of 2.8 billion euros, and we have about 30% as founded by the German federal and state governments, and more than 70% is self-acquired by contracts with industry and from publicly financed research projects. The institute I'm working at is located in the beautiful Brandenburg in Potsdam. And Potsdam is nowadays not only known for the Palace of Sanssouci, but also for the Potsdam Science Park, where we have 2 Fraunhofer institutes and several Max Planck institutes, the University of Potsdam, and a lot of companies. The Fraunhofer ECBB is focused on bioanalytics and bioengineering, and the Department of Cell-Free and Cell-Based Bioprotection contributes to the bioengineering part. We had already a good introduction to click chemistry, so I just want to give some more examples to it. So most of the people want to stain their target proteins, and often they're using fusion proteins like GFP, or they use monoclonal antibodies. And even in particular, if you have small target proteins, the GFP or the monoclonal antibody might have a really high molecular weight and might influence the structure and the function of the target protein. So therefore, we need small protein labels, and really common labeling strategy is to click these small molecules via click chemistry. As was already mentioned, there are a lot of applications for click chemistry, and I want to focus today on the protein modification and immobilization, and on the conjugation and labeling of biomolecules. So these biomolecules are proteins, and usually proteins are produced in vivo protein production system. These are based on prokaryotic systems such as Escherichia coli, or on eukaryotic systems like yeast, insect cells, and mammalian cells. Each of the system has some advantages and disadvantages. So the prokaryotic systems usually have high protein yields and low cultivation costs, but they are limited in post-translational modifications, such as glycosylation, that are for some proteins really important for their function. So eukaryotic systems have high cultivation costs, but usually they are able to really produce active, complex, and high molecular weight proteins. But all of these in vivo protein production systems have some general disadvantages. In particular, if you want to try to produce toxic proteins or proteins that have cytotoxic effects. So in particular, membrane proteins, if we try to overexpress them and they start signal cascades, these signal cascades might end the death of the cell. Moreover, as I mentioned, cell culture is time-consuming and often related to high costs. Therefore, our group is focusing on the development of alternative protein production systems, and here comes the cell-free protein synthesis system. First of all, we start as well with the cultivation of cells, and there we are using batch perfusion processes. If we have a particular cell density, we harvest our cells by centrifugation, we obtain the cell pellet, and then we disrupt our cells. So this disruption method is a really mild disruption because we want to obtain the inner life of the cell in an active state. So, and therefore we gain the complete translational machinery, and then we supplement this and we do a chromatography step, and what we obtained is a so-called lysate. And in this lysate, you can see it here on the left side, we have also components that are necessary for protein translation that is independent of a living cell. So we have the ribosomes, we have the translation factors, we have the tRNAs, and the aminoacylTRN synthetase. So to this lysate, we add some factors such as a DNA template, where we have the genetic information for our protein of interest. This can be circular or linear. We add amino acids, energy, and an energy regeneration system. Also supplements such as salts. An advantage of this system is that we add undefined concentration of amino acids, and we add on 14C leucine to the system. Since this amino acid is radio-labeled, we can do SDS page followed by an autoradiography to really see the de novo synthesized protein that has statistically incorporated 14C leucine. Moreover, with this procedure, we can determine the protein yield of the de novo synthesized protein. As I mentioned, we are working with eukaryotic cell-free systems, and this system has one advantage, and this is the presence of so-called microsomes. These microsomes are membrane vesicles that are derived from the endoplasmatic reticulum, and the endoplasmatic reticulum is obtained during the mild disruption procedure and is as well an active form, so there is a translocon present, and the translocon enables the co-translationally translocation of secreted and membrane proteins. So, and with this translocation, we have as well post-translational modifications such as glycosylation and disulfide bridging. Moreover, the synthesis reaction I have described here takes place in 3 to 24 hours. And we have an open system, and the open system enables the addition of exogenous components. And such components can be modified synthetases, modified tRNAs, and non-canonical amino acids. And with this basic, we can use the methodology of AMPA suppression to incorporate a non-canonical amino acid at a specific position in our protein of interest. So how does it work? First of all, we start as well with an DNA template with the genetic information, and we introduce into the gene sequence an AMPA stop codon at the desired position. Usually, the protein translation would stop at the AMPA stop codon, and we would obtain a so-called termination product, so a truncated protein. But since we have added a modified synthetase that can recognize the modified amino acid and is able to load the unnatural amino acid to the modified tRNA, the tRNA has a complementary codon to the AMPA stop codon, and as a result, the non-canonical amino acid is connected to the polypeptide chain doing the protein translation. So that means instead of the truncated protein, we get the full-length protein, but we have the non-canonical amino acid with a reactive group at the desired position in our protein. So that this happens specific as we want. The endogenous synthetases are not allowed to cross-react with the modified tRNA, and the endogenous tRNAs are not allowed to cross-react with the exogenously added synthetase. The non-canonical amino acids we are using are usually modified phenylalanines. So as you can see here, one of the phenylalanines bears an acid group, and the reactive acid group can then further be modified via Staudinger ligation, kappa catalyzed cycloaddition, and strain promoted cycloaddition. So as I mentioned, we are focusing mainly on post-translational modified proteins and therapeutical proteins. And one of the most famous proteins in this part is glycoprotein erythropoietin, short EPO. And EPO was till 2004 one of the 10 most successful medicals. And moreover, there are a lot of biosimilars of EPO because it has an immense therapeutical effect for ischemia and to regulate the oxygen in our blood. So, and a lot of researchers are interested in a really defined EPO with defined glycosylation pattern. So about 40% of the molecular weight of EPO is composed of the glycosylation. Usually, if you have an in vivo production process, you have heterogeneous glycosylation. So even though you have glycosylation, the small molecules of the sugar are different between molecules and in between the same molecule. So, and therefore, they are focusing on a method to obtain an EPO that has a lot of glycosylation, because the hyperglycosylated EPO has a higher half-life and an increased in vivo bioactivity, but to obtain defined glycosylation pattern. And therefore is EPO one of the most glyco-engineered protein in the last years. And we thought with our cell-free protein synthesis, we might be able to engineer as well the erythropoietin. So not with a defined sugar moiety, but with polymer. And therefore, we started again to introduce an AMPA stop codon into the gene sequence of EPO. We have chosen the position where we usually would expect the O-glycosylation, because O-glycosylations are not possible in cell-free systems. And then we have determined if we can incorporate different non-canonical amino acids. You can see on the left side, again, the acid-modified phenylalanine, and on the right side, you can see the alkyne-modified phenylalanine. We have checked the incorporation of the non-canonical amino acids via the coupling to the fluorescent dye. So the AZF was coupled to a dilute phosphine, and in the in-real fluorescence, we can see a specific band pattern. And Erythropoietin has 3 glycosylation sites. So what we can see here is the smallest band is the unglycosylated EPO. Then we have the 1-time glycosylated EPO, 2-time and 3 times glycosylated EPO. If we have now an glycosidase digestion, we see that the glycosylation pattern are disappearing and we have only the fluorescent band of the deglycosylated full-length EPO in our NG fluorescence. As well, you can see if we do not use one of the orthogonal component, as I have mentioned, we have the synthetase, the exogenous tRNA, and the non-canonical amino acid, we see that there is not a misincorporation at the AMBA stop codon. So because what you can see on the autoradiograms, so here we have our radioactively labeled erythropoietin, you can see if we add all components, we get the specific band pattern. If we do the glycosylation digestion, we see that we have the termination product deglycosylated and the full-length product deglycosylated. If we now add not the non-canonical amino acid, we only get the deglycosylated termination product. So here we can see that we have no misincorporation at the AMBA stop codon. So as I mentioned, the goal was to specifically modify the EPO with the biopolymer. As a biopolymer, we have chosen an polyethylene glycol. This one was modified with an S-side group. And via the couple catalyzed click reaction, we have then coupled the PEG to our alkyne group of the incorporated phenylalanine. And you can see here at the band of 49 kilodalton, we have a specific band pattern that is new. And this band corresponds to the fully glycosylated EPO that is coupled to our biopolymer PEG. Of course, now it is interesting if the self-resynthesized protein shows comparable activity to non-self-resynthesized proteins. And therefore, we have performed an cell culture assay. Here you can see an OT7 cell line. The cell line is sensitive to growth hormones such as EPO. And on the left side, you can see there's the black line where we have the cell-free EPO, and we have the dotted line where we have the commercially available EPO of sigma. And both lines are relatively comparable. So we see here that our self-resynthesized protein shows nearly the same activity at the commercially available EPO. This experiment was performed with a non-modified EPO, but new experiments, they are not yet published, show that we have as well an active EPO if we couple the EPO to biopolymers such as the mentioned PEG. As I mentioned in the beginning, we are focusing as well on membrane proteins, and one large category that is really interesting are GPCRs. And one of a famous GPCR, the adenosine A2A receptor, because this receptor is involved in diseases such as Parkinson's disease. It controls the immune function, and it is involved in the cardiac rhythm and circulation. Our goal was here to see if the proteins that are synthesized in our cell-free system show similar behavior than the proteins that are synthesized in a living cell. And in particular for this receptor, there's a unique feature described. So in an inactive form, you can have an bioluminescence resonance energy transfer between a C-terminally fused nanoluciferase and a fluorophore that is introduced into the third intracellular loop of the GPCR. So again, therefore, we have introduced the AMBA stop codon into the gene sequence that would result in an amino acid located on the third intracellular loop, and we have C-terminally fused nanoluciferase. Afterwards, we have coupled the fluorescent dye to the non-canonical amino acid. So as you can see on the left side, as I have described, we would expect in an inactive state, we would have an energy transfer between the nanoluciferase and the fluorophore. If we don't add any ligand, so 0 micromolar, we see we have already a signal. That's what we expected. And if we now add adenosine, we would expect that there's a dramatic change in the transmembrane domains of the receptor that would result in an proximity that is different to the proximity between the luciferase and the fluorophore in the inactive state. And you can see on the right side that if we add a ligand, the ratio between the luminescence and fluorescence is changed. And therefore, we can determine that we have a ligand-dependent conformational change of our receptor in the cell-free synthesized system. Last but not least, I want to speak about antibodies, since antibodies are as well a really, really famous group of proteins. Everybody knows for therapeutical issues and for analysis. And in our cell-free system, we have as well synthesized different formats of antibodies. So we have the full-length antibody consistent of 2 heavy chains and 2 light chains called IgG. Then we have the single chain Fc format that is consistent of the heavy chain and the single chain fragment. Then we have the single chain fragment, and we have the VHH. On the left, you can see again the autoradiogram where we have incorporated the 4-C leucine into our de novo synthesized protein, and we get a specific band for the single chain fragment. We get a specific band for the light chain. For the heavy chain, we see the monomer and we see the dimeric heavy chain. And if we now combine the light chain with the heavy chain, we see that we have additionally to the light chain and heavy chain that we get as well a band that corresponds to the assembled full-length antibody. The antibody can as well be purified and then applied to an activity assay, or let's say to SPR analysis, where we have seen a specific binding signal of the antibody and of the single chain fragment Fc construct. The antibody was then used for a specific labeling, and therefore we have again used our cell-free synthesis. We have introduced the AMA subcodon at the position where the antibody is not changed in its function, and we have incorporated the acid reactive amino acid and have then coupled the fluorescent dye. And we see again the different formats of the synthesized protein. We have the monomeric heavy chain, we have the dimeric heavy chain, we have the dimeric heavy chain with 1 single chain, and last but not least, we have the full-length antibody comprising of 2 light chains and 2 heavy chains assembled automatically in a cell-free system. As well, this assembly is only possible since we have the possibility to have post-translational modification such as disulfide bridging in our cell-free system. With this example, I want to finish my talk and hope I could show you several applications for click chemistry and combinations with a cell-free protein synthesis. And with this, I want to thank the whole Department of Cell-Free and Cell-Based Bioproduction, our financial support, and of course, IRIS Biotech for giving me the possibility to present our results here. And I would be happy if you have any questions. Thank you.
Thank you, Anne, for sharing your results with us. And we already have several questions within the chat, so I just start out with the first one. So this one is, do you need to purify your proteins before applying to the specific application?
This depends on the application. So from the 3 examples I have shown you, only the antibody was purified for the SPR measurement. So for the cell culture assay, we have directly applied our cell-free reaction. So usually the translation mix or the cell-free reaction has no influence on cell culture or on activity assays, because luckily nowadays a lot of activity assays are so specific that even though we have a mixture of protein, only the active protein of interest is focused on the assay.
Okay, thanks. So that's actually facilitating your work a bit, right?
Yes, it does.
Okay, and can you comment on typical protein yields And also the question is the system scalable?
So the protein yields highly depend on the system we are using. You might have seen that there was a small picture of protein yields with the antibody slides. And we have two possibilities. So we have the batch reaction that is really in an Eppendorf tube. The reaction is only 3 hours. So there we have protein yields in the, let's say, 20 to 50 microgram per milliliter. So the alternative is to use dialysis system. Here we have a 2-chamber system, and the reaction chamber is permanently feeded with amino acids, energy, and salts. And here we have a prolonged reaction lifetime that can be up to 24 or even 48 hours. With this reaction, we can reach up to 1 milligram per milliliter. So that's comparable to 1 gram per liter. And with this protein yield, we are comparable to in vivo protein production. And the scalability, the work I have shown you was mainly performed in a microliter scale, but in principle, it is possible to scale up the system. For an E. coli cell-free system, there's one publication where they have done the synthesis in 100 liter. Usually we are not doing it in 100 liter, but let's say to 5 to 10 milliliter is possible to scale it up.
Okay, that was really a number I didn't expect, 100 liters.
Yeah, there you have really these large wave bags where you have the cell-free reaction in it.
Great, so thanks for your detailed answer. One more question is, can you comment on the incorporation efficiency of the non-canonical amino acid?
Yes. So since our system is based on the natural living cell, even though we are not using the living cell, we have as well translation factors and enzymes and release factors in it. So, and if we want to incorporate our non-canonical amino acid, we have always a competition between the non-canonical amino acid and the release factor 1 that will end the protein translation at the AMBA stop codon. So, but for the examples I have shown you, usually our incorporation efficiency is about 70 to 90%. So we have a really good incorporation efficiency. So, but usually this is related because we can really add defined concentrations of the tRNA, the non-canonical amino acid, and the synthetase. And therefore we can get the best combination of all 3 components to really have a high incorporation efficiency of the non-canonical amino acid.
Okay, great, thanks. And one last question so far. So is it possible to introduce more than one non-canonical amino acid into one protein?
It is. So there, I would say we have 2 options. So what we could do is to install 2 AMBA stop codons into the gene sequence. So then we would introduce the same non-canonical amino acid at 2 positions in our protein. But of course, we will get the same problems, also the release factor I have explained. And there we will get a lower incorporation efficiency, maybe about 50%. But what we could do as well is to use some second orthogonal systems, so a second synthetase, a second non-canonical amino acid, and a second tRNA that are addressing a different stop codon. And then we could incorporate 2 different non-canonical amino acids at each defined position.
Okay, so I just see that also regarding this question, there was one answer by an attendee and he recommends one publication. So for the one anonymous, he was interested in this question. Please, maybe you can also check out the publication, and thanks to Raimund for sharing this answer with us. And also thanks to Anne, of course, for answering the questions. So I can't see any further questions in the chat. Sometimes it takes a while until they appear, but of course we are also willing to answer your questions afterwards. So just directly contact us and we will help you with your questions. So Anne, thanks again. It was a pleasure to have you as our external guest speaker.
Thank you.
And Yeah, no further question. There was one more question to Markus within the chat. There was just a slight delay in time, but Anamika, as you might see, Markus already answered in the chat, so I hope that this one is also clear. So yeah, with this, I would say we would like to close our online workshop just in time. As a follow-up, of course, we will provide the recordings and a collection of the Q&A as PDF. I would like to thank you all again for joining us. Thanks again to all our speakers, to Steffen, to Markus, to Anne, and also a very big thanks to Dr. Klaus Helm. So he was not visible, but he took care of all the technical support. And yeah, we hope you had as much fun as we had during this first online workshop. And we are looking forward to e-meeting you again in the next one. So just stay tuned and we will announce it on time on our homepage and also on social media. So with this, have a nice day and see you soon in the next workshop.